Chapter 20 Transports in Plant. Plants need to move molecules over very long distances, much more than animals do, and they also do not have a circulatory system in place. Water taken up by roots has to reach all the parts of the plant up to the very tip of the growing stem. The photosynthesis or food synthesized by the leaves also to be moved to all parts, including the root tips embedded deep inside the soil. Movements across short distances, say within the cell, across the membranes, the from cell to cell within the tissue has also takes place. To understand some of the transport processes, we must know that structure of the cell, anatomy of the plant body, process of diffusion, and the chemical potential and ions. In a flowering plant, the substances that would need to be transported are water, minerals, nutrients, organic nutrients, and plant growth regulators. Over small distances, substance moves by diffusion and by cytoplasmic streaming, supplemented by active transport. Transport over long distances proceeds through the vascular system, xylem and phloem, which is called translocation. The bulk movement of substances through the conducting or vascular tissue of plant is called translocation. In rooted plants, transport in xylem of water and minerals is essentially unidirectional from roots to the stem. Direction of movements in phloem of organic and mineral nutrients is bidirectional and multidirectional. Hormones or plant growth regulators and other chemical stimuli are also transported through, though in very small amounts, sometimes in a strictly polarized or unidirectional manner from where they are synthesized to other plants. Means of transport 1. Diffusion. Movement by diffusion is passive, that is, in diffusion, no energy expenditure takes place, no ATP utilized. By diffusion, movement may be from one part of the cell to another, or from cell to cell, or over shorter distances, say from intercellular spaces of leaves to the outside. By diffusion, movements may be from one part of cell to another, or from cell to cell, or over short distances, say from intercellular spaces of leaf to the outside. In diffusion, molecules moves in a random fashion, the net results being substances moving from region of higher concentration to region of lower concentration, or along the concentration gradient. Diffusion is a slow process and is not dependent on a living system. Diffusion is obvious in gases, liquids, but diffusion in solids rather than of solids is more likely. Uniformly sweet taste due to tea or coffee is due to diffusion. Diffusion is very important to plants because it is only means of for gaseous exchange within the plant body. Diffusion rates are affected by concentration gradient, pressure, membrane permeability, and temperature. Facilitated diffusion. Substances that have a hydrophilic moiety find it difficult to pass through the membrane along the concentration gradient, so their movements has to be facilitated. Membrane proteins provide site at which such molecules cross the membrane. This process is called facilitated diffusion. Membrane proteins do not set up a concentration gradient. A concentration gradient must already be present for molecules to diffuse, even if facilitated by the proteins. In facilitated diffusion, special carrier proteins have helped move substances across membranes without expenditure of ATP energy. Transport rate reaches maximum when all the protein transporters are being saturated, used. Facilitated diffusion is very specific. It allows cells to select substances for uptake. Facilitated diffusion is sensitive to inhibitors which react with the protein chain. Proteins form channel in membrane for molecules to pass through. Some channels are always open, others can be controlled. Some are large, allowing a variety of molecules to cross. The porins are proteins that form huge pores in the outer membrane of the plastids, mitochondria, and some bacteria, allowing molecules up to the size of small proteins to pass through. Figure 1 shows an extracellular molecule bound to the transport protein. The transport protein then rotates the release in the molecule inside the cell. Example: water channels is made up of 8 different types of aquaproteins. Some carrier or transport proteins allow diffusion only in two types of molecules or move together. If both the molecules cross the membrane in the same direction, symport. If both the molecules move in opposite direction, antiport. When molecules move across a membrane independent of other molecules, uniport. Diffusion rates depends on the size of substances. Smaller molecules diffuse faster. Diffusion of any substance across a membrane also depends on its solubility. Substances soluble in lipids diffuse through membrane faster. Active transport. Active transport uses energy to pump molecules against the concentration gradient. Active transport is also carried out by membrane proteins. Pumps are proteins that use energy to carry substances across the cell membrane. These pumps can transport substances from a low concentration up to a high concentration, uphill transport. Like enzymes, the carrier protein is very specific. Uniport, antiport, symport. These proteins are sensitive to inhibitors that react with protein side chains. Proteins in the membrane are responsible for active transport and facilitated diffusion. Passive transport, therefore, they show common characteristics. Highly selective, liable to unsaturated respond to inhibitors under hormonal regulation, but diffusion, whether facilitated or not, takes place only along a gradient and do not use energy. Property requires special membrane proteins. Simple diffusion, no. Facilitated diffusion, yes. Active transport, yes. Highly selective, no. Yes, yes. Transport saturates, no. Yes, yes. Uphill transport, no, no, yes. Requires ATP, no, no, yes. Rand water relationships. Plant-water relationships. Water is essential for all physiological activities of the plant and always plays a very important role in all living organisms. Water provides the medium in which most substances are dissolved. The protoplasm of the cell is nothing but water in which different molecules are dissolved and several particles are suspended. A watermelon has over 92% water. Herbaceous plants only have 10 to 15% of its fresh dry weight as dry matter. Of course, distribution of water within a plant varies. Woody parts have relatively very little water, while soft parts mostly contain water. A seed may appear dry but it still has water, otherwise it would not be alive and respiring. plants take up huge amount of water daily, but most of it is lost to the air through evaporation from the leaves, that is transpiration. A mature currant plant absorbs almost 3 liters of its water in a day. A mustard plant absorbs equal to its own weight in about 5 hours. Because of this demand for water, it is not surprising that water is often the limiting factor for plant growth and productivity in both agriculture and natural environments. Water potential, solute potential Ys and pressure potential Yp are the two main components that determine the water potential. Water potential is a concept fundamental to understanding water movements. The term water potential was given by Slater and Taylor. The greater the concentration of water in a system, the greater is its kinetic energy of the all water potential. Pure water will always have the greatest water potential. Water will move from system containing water at higher water potential to one having low water potential. This process of movement of substances down a gradient of free energy is called diffusion. Water potential is denoted by Greek symbol psi and is expressed in pressure units such as Pascal. By convention, water potential of pure water at standard temperature, which is not under any pressure, is taken to be zero. If a pressure greater than the atmospheric pressure is applied to a pure water or a solution, its water potential increases. It is equivalent to pumping water from one place to another. If some solid dissolved in pure water, then the solution has fewer free water and concentrations of water decreases. Hence, all solutions have a lower water potential than pure water. The magnitude of lowering in water potential due to the dissolution of solute is called solute potential. Solute potential is always negative. For a solution at atmospheric pressure, water potential is equal to solute potential. As compared to 1 molar sucrose or glucose solution, water potential of the solution of the 1 molar sodium chloride solution will be lower. Pressure exerted by the chloroplast to the entry of water against the rigid wall is called pressure potential. Due to diffusion of cell water into the cell, cytoplasm builds up a pressure against the wall. It is called turgor pressure. Turgor pressure develops in epiphyllema cell of the root due to entry of water into root hair and increase in volume of the cell sac. Turgor pressure is ultimately responsible for enlargement and expansion of the growth of cells. Pressure potential is usually positive, but in plants, negative potentials or tension in the water column in the xylem plays a major role in water transport upper stem. Water potential of a cell is affected by both solute and pressure potential. DPD, diffusion pressure deficit, is also called suction pressure. DPD is equal to OP minus DP. Question: cells A and B have contiguous. Cell A has OP 10 atmosphere, TP 7 atmosphere, and DPT 3 atmosphere. Cell B has OP 8 atmosphere, TP 3 atmosphere, and DPT 5 atmosphere. The result will be no movement of water, equilibrium between two, movement of water from A to B, movement of water from B to A. DPT becomes more than OP if TP is negative. If turgor pressure becomes equal to osmotic pressure, then no exchange of water takes place. OP of a vacuolated plant cell is less than DPT. Water movement is from lower DPT to higher DPT. DPT becomes more than OP if TP is negative. If turgor pressure becomes equal to the osmotic pressure, then no exchange of water takes place. OP of a vacuolated plant cell is less than DPT. Water movement is from lower DPT to higher DPT. Osmosis. The plant cell is surrounded by a cell membrane and a cell wall. Cell wall is freely permeable to water and substances and solution, therefore is not a barrier to movement. In plants, the cells usually contain a large central vacuole whose contents, the vacuole or sap, contribute to the solute potential of the cell. In the plant cells, the cell membrane and chloroplast membrane of vacuole together are important determinants of movements of molecules in or out of the cell. Osmosis is a special type of diffusion. Osmosis is diffusion of water across a differentially or semi-permeable membrane. Semi-permeable allows the diffusion of only solvent. Semi-permeable membrane may be membrane of egg. Remove the yolk and albumin through a small hole at one end of egg. Place the shell in dilute solution of HCl for a few hours. The egg shells dissolves, leaving membrane intact. The net direction and rate of osmosis depends on both pressure gradient and concentration gradient. Water will move from its region of higher water potential concentration to its region of lower water potential, or from less concentrated solutions to more concentrated solutions until equilibrium is reached. At equilibrium, the two chambers should have the same water potential. There are two chambers A and B. A have lower solute molecule than B. Then, solution of which chamber has lower water potential, direction in which osmosis occurs. Okay, this is an important example. You can try it with yourself. If a chamber Y has okay, this is again an example. In plants, water moves from less negative to more negative gradient. Supply of excess fertilizers and watering of a grass lawn causes browning of grass leaves due to osmosis and death of roots. Demonstration of osmosis is done by thistle funnel or potato osmometer. Potato osmometer: if the tuber is placed in water, the cavity in the potato tuber containing a concentrated solution of sugar collects water due to osmosis. Thistle funnel experiment: a solution of sucrose in water taken in a funnel is separated from pure water in a beaker through a semi-permeable membrane. Water will move into the funnel, resulting in the rise in the level of solution in the funnel. This will continue till equilibrium is reached. External pressure required to prevent water from diffusing in thistle funnel is called osmotic pressure. If sugar is added in a beaker after the equilibrium is reached, then level of solution in thistle funnel lowers. Osmotic pressure is maximum in halophytes. Osmotic pressure is the function of solute concentration. More of the solute concentration, greater will be the pressure applied to prevent the water from diffusing it. Osmotic pressure of a solution is more than that of pure solvent. A demonstration of osmosis: a thistle funnel is filled with sucrose solution and kept inverted in a beaker containing water. Water will diffuse across the membrane as shown by arrow to raise the level of solution in the funnel. The pressure can be applied as shown to stop the water movements into the funnel. A demonstration of osmosis: a thistle funnel is filled with sucrose solution and kept inverted in a beaker containing water. Water will diffuse across the membrane as shown by arrows to raise the level of the solution in the funnel. Pressure can be applied as shown to stop the water movement into the funnel. Numerically, osmotic pressure is equivalent to osmotic potential, but the sign is opposite. Osmotic pressure is positive pressure, while osmotic potential is negative. Osmotic pressure potential of pure water is 0. Plasmolysis: if the external solution balances the osmotic pressure of cytoplasm, it is said to be isotonic. If the external solution is more dilute than the cytoplasm, it is hypotonic. If the external solution is more concentrated, it is hypotonic. Cell swells turgid in hypotonic solution and shrinks plasmolite in hypertonic ones. When the water flows from cell and out of the cell are in equilibrium, the cell is said to be flaccid. When a cell is fully turgid, water potential dPd suction pressure is 0. When RBC and a plant cell having same OP are placed in distilled water, then RBC swells up and bursts, but the plant cells remain about the same size. When cell is placed in hypertonic solution, plasmolysis occurs. Water moves out of the cell and cell membrane of the plant cells shrink away from its cell wall. If a slice of sugar beet is placed in concentrated salt solution, then it would lose water and become plasmolite. A cell when dipped in 0.5 molar sucrose solution has no effect, but when the same cell is dipped in 0.5 molar NaCl solution, it will get plasmolized. Water that moves out exosmosis is first lost from cytoplasm and then from the vacuole. Hypertonic solution occupies the space between the cell wall and the shrunken protoplast in the plasmolite cell. Bacteria do not survive in highly salted pickle because they become plasmolized and then killed. Salt, sugar, and vinegar preserves food stuff. The process of plasmolysis is usually irreversible. When the cell are placed in a hypertonic water solution, water diffuses into the cell, causing its swelling. Because of the rigidity of cell wall, the cells do not rupture. Excessive supply of chemical fertilizers often cause wilting or death of crops or plant due to exosmosis. Resins placed in water swell up due to endosmosis. Imbibition. Imbibition is a special type of diffusion. In imbibition, water is absorbed by solids and increased in volume. Classical example of imbibition is absorption of water by seeds and dye wood. The pressure that is produced by the imbibition is used by the prehistoric men to split rocks and boulders. Due to the pressure of imbibition, seedlings would be able to emerge out of the soil into the open. Imbibition is also diffusion because water movement is along concentration gradient. The seeds and other such materials have almost no water, hence they absorb water easily. Water potential gradient between the absorbent and the liquid imbibed is essential for imbibition. For any substances to imbibe any liquid, affinity between the absorbent and the liquid is also a prerequisite. Agar is the most efficient imbibant. Long distance transport of water. Long distance transport of water. At some earlier stage, you might have carried out an experiment where you had placed a twig bearing white flowers in colored water and had watched it turn a color. On examining the cut end of the twig, after a few hours, you had noted the region through which the colored water moved. This experiment very easily demonstrates the path of water movement is through the vascular bundles, more specifically the xylem. Long distance transport of substances within a plant cannot be diffusion alone. Diffusion is a slow process. It can occur only for short distance movement of molecules. Movement of molecule across a typical plant cell, about 50 micrometer, takes approximately 2.5 seconds. In large and complex organisms, often substances have to be moved across very large distances. Sometimes the sites of production or absorption and the sites of storage are too far from each other. Diffusion or active transport would not suffice a special long distance transport system because necessary so as to move substances across long distances and at a much faster rate. Water, mineral, and food are generally moved by mass or bulk flow system. Mass flow is a movement of substances in bulk or in mass from one point to another as a result of pressure differences between the 2 points. It is a characteristic of mass flow that substances, whether in a solution or in suspension, are swept along at the same pace as in a flowing river. This is unlike diffusion, when different substances move independently, dependent on their concentration gradient. Bulk flow can be achieved either through positive hydrostatic pressure gradient, garden hose, negative hydrostatic pressure gradient, suction through a straw. Xylem is associated with translocation of mainly water, mineral salts, some organic nitrogen, and hormones from root to shoot. Phloem translocates a variety of organic and inorganic solids from the plant, from the leaves to the other parts of the plant. Absorption of water. We know that the roots absorb most of the plants that goes into plants. Obviously, that is why we apply water to the soil and not on the leaves. The responsibility of absorption of water and the mineral is a function of root hair and are present in the zone of maturation. Root hairs are thin-walled, slender extension of root epidermal cells that greatly increase in surface area for absorption. Water is absorbed along with mineral solids by root hair purely by diffusion due to suction pressure or osmosis. Transpiration in leaves is the driving force for the process of passive absorption of water in root. Water potential of root cell is lower than water potential of soil. Water absorption by root hair occurs until water potential is lower. When concentration of solution of low in the soil, absorption of water is increased. Once water is absorbed by root hair, it can move deeper into root layers by two distinct pathways: apoplast pathway and synplast pathway. Apoplast is a system of adjacent cell walls that is continuous throughout the plant, except at the Caspian strips of the endodermis of root. The apoplastic movement of water occurs exclusively through the intercellular spaces and the walls of the cell. Movements through the apoplast does not involve crossing the cell membrane. Apoplastic movement is dependent on the gradient. The apoplast does not provide any barrier to water movement, and water movement is through mass flow. Mass flow of water occurs due to adhesive and cohesive properties of water. Mass flow of the water flows in the root occurs via the apoplast since the cortical cells are loosely packed. Hence offer no resistance to water movement. However, the inner boundary of cortex, that is endodermis, is impermeable to water because of a band of suberized matrix called the Caspian strip. Water molecules are unable to penetrate the layer, so they are directed to wall regions that are not suberized into the cell proper through the membrane. If a plant cell covered with cutin and suberin is placed in water, then after 15 minutes the cell will remain unchanged. Water then moves through the symplast and again crosses the membrane to reach the cell of the xylem. The movement of water through the root layers is ultimately symplastic in endodermis. This is the only way of water and other solutes can enter the vascular cylinders. Pathway of water movement from soil to xylem is soil, root, hair, cortex, endoderms, pericycle, protoxylem, metaxylem. Ascending order of DPD is the root hair, cell, cortical cell, and mesophyll cell. Ascending order of DPD is root hair, cell, cortical cell, and mesophyll cell. The movement of water from one cell of cortex to the next in the roots is due to water partitioning gradient (DPD). Symplast pathway. Terms apoplast and symplast were first used by Munch. Symplastic system is the system of interconnected protoplasts. Neighboring cells are connected to plasmolysis. During symplastic movement, the water travels through the cell by the cytoplasm. Intercellular movement is through the plasmolysis. In symplast, water has to enter the cells through the cell membrane, hence movement is relatively slower. Symplastic movement by the aided cytoplasmic streaming. Cytoplasmic streaming observes in cells of Hydra leaf. The movement of chloroplast due to the streaming is easily visible once inside the xylem. Water is again free to move between cells as well as through them. In young roots, water enters directly into the xylem vessel or tracheids. The xylem vessels and tracheids are the non-living conduits channel, and so they are part of apoplast. Some plants have additional structures associated with them that help in water and mineral absorption. Mycorrhiza is a symbiotic association of a fungus with the root system. The fungal filament forms a network around the young root or they penetrate the root cells. The hyphae have a very large surface area that absorbs mineral iron and water from the soil from a much larger volume of the soil that perhaps a root cannot do. Fungus provides water and minerals to the root and root in turn provides sugar and N-containing compounds to mycorrhizae. Some plants have an obligate association with the mycorrhizae. For example, pinus seeds cannot germinate and establish without the presence of mycorrhizae. This is an example of obligate association. Water movement up a plant. We looked at how a plant absorbs water from the soil and move it into the vascular tissues. We now have to try and understand how this water is transported to various parts of the plant. Root pressure. As the various ions from the soil are actively transported into vascular tissues of the root, water follows its potential gradient and increases the pressure inside the xylem. This positive pressure is called root pressure and can be responsible for pushing up water to small heights in the stem. Force developed in cortex of root which pushes water into xylem of root is called root pressure. How can we see that root pressure exists? Choose a small soft stem plant and on a day when there is plenty of atmospheric moisture, cut the stem horizontally near the base. With a sharp blade early in the morning, you will soon see drops of solution oozing out of the stem cut. This comes out due to the positive root pressure. If you fix a rubber tube to the cut stem as a sleeve, you can actually measure the rate of exudation and also determine the composition of exudates. The effects of root pressure is also observable at night and early morning when evaporation is low and excess water collects in the form of droplets around special opening of veins near the tips of the grass blades and leaves of many herbaceous parts of plant. Topolium, balsam, and grasses. Such water loss in its liquid phase is known as guttation. Guttation is mainly due to positive root pressure. Guttation is elimination of water through hydrothotes. Hydrothotes remains always open. Root pressure is maximum at the time when absorption is high and transpiration is low. Water potential in leaf tissue is a positive near zero during guttation. Root pressure can provide only a modest, simple push in the overall process of the water transfer. The greatest contribution of root pressure may be to re-establish the continuous chain of water molecules in the xylem, which often break the under-enormous tension created by transpiration. Root pressures do not account for the majority of water transport. Most plants meet their need by transpiration pull. Oozing out of water drops from inside edge or tips is bleeding. Transpiration pull. Despite the absence of a heart or a circulatory system in plants, the flow of water upwards to the xylem can achieve fairly high rate up to 15 meters per hour. How is this movement accomplished? A long-standing question is put whether water is pushed or pulled to the plants. Most researchers agree that the water is mainly pulled through the plant and driving force for this process is transpiration from the leaves. This is referred to as cohesion tension transpiration pull model of water transport, which operates in passive absorption. The most widely accepted theory for the extent of sap is transpiration pull by Dixon and Johnny. For extent of sap, capillary force theory was first proposed by Christian Wolff. Pulsation theory of ascent of sap was given by J.C. Bose, while relay pump theory was given by Godlewski. Less than 1% of water reaching the leaves is used in photosynthesis and a plant growth. Rest 99% of water is lost in transpiration. Most of the water absorbed is lost through the stomata in the leaves. This water loss is known as transpiration. You have studied transpiration in an earlier class by enclosing a healthy plant in polythene bag and observing the droplets of water formed inside the bag. You could also study the water loss from a leaf using cobalt chloride paper, which turns color on the absorbing water. Cobalt chloride paper is used for determining differential transpiration or unequal transpiration on two surfaces. Dry cobalt chloride is blue while the moist is pink in color. A leafy twig of mesophytic plants dipped in water would demonstrate transpiration. Fourth, transpiration. Transpiration is the evaporative loss of water by plants. Transpiration occurs mainly through the stomata in the leaves. Transpiration is mainly a process of diffusion. Aquatic submerged plants do not show transpiration. Hydrilla has non-functional stomata. Transpiration differs from evaporation in transpiration, a physiological process, while evaporation is a physical process. Water lost through transpiration is pure water. Rate of transpiration is determined by potometer or Ganong's potometer. Atmometer or evaporimeter is used for measuring the rate of evaporation of water from a wet surface to the atmosphere. Blue and red region are the extent spectrum of transpiration. About 10,000 stomata are present per centimeter square of a leaf. Besides the loss of water vapor in transpiration, exchange of O2 and CO2 in the leaf also occurs through stomata. Normally, stomata are open in daytime because the guard cells have chlorophyll but close during night. But in cam plants, stomata close during day and open in night to reduce the loss of water to transpiration in xenic habitat. Types of stomata. Potato type, a leaf with more stomata on lower surface. Water lily type, leaf with stomata present exclusively on upper surface. Barley type, stomata open only for a few hours during the day. Example, cereals. Alfalfa type, stomata remains open throughout the day and closed during the night. Immediate cause of opening and closing of stomata is a change in turgidity of guard cells. Guard cells are unique cells which are fewer in number and chlorophyllous. Common between guard cells and mesophyll cells is the presence of chloroplast. Guard cells differ from epidermal cells in having chloroplast. Inner wall of each guard cell towards the pore or stomatal aperture is thick and elastic. When turgidity increases within the two guard cells, the thin outer walls bulge out and force the inner wall into a crescent shape and stomata becomes open. Stomata open in daytime due to decrease in water potential. The opening of stomata is aided due to orientation of cellulosic microfibrils in the cell wall of the guard cells. Cellulose microfibrils are oriented radially rather than longitudinally, making it easier for the stomata to open. When guard cells lose turgor due to water loss or water stress, the elastic inner wall regains their original shape. The guard cells become flaccid and the stomata closes. High CO2 concentrations in leaf interior will cause stomatal closure. Decrease in CO2 concentrations and decrease in H plus ion concentrations causes opening of stomata. A sudden increase in CO2 concentration around the leaf will cause decrease in transpiration due to closure of stomata. Active K plus ion exchange mechanism for the opening and closing of stomata was given by Levitt. Efflux of KCl and influx of H plus ion causes closure of stomata. ABA is produced during water stress that brings stomata closure. Cytokinins open stomata. Stomata open when guard cells possess more K+. Starch, stomatal opening, organic sugars, stomatal closing, starch. Stomata open during day due to accumulation, influx of K plus ion and sugar. Starch. ABA theory is the latest explanation for closure of stomata. Pep carboxylase is connected with opening and closing of stomata. Usually, the lower surface of a dorsiventral dicotyledonous leaf has a greater number of stomata. On upper surface, stomata may be even absent sometimes. In isobilateral monocotyledonous leaf, stomata are about equal on both surfaces. Transpiration is affected by several external factors: temperature, light, humidity, wind speed. Transpiration is least in high atmospheric humidity or relative humidity. Due to excessive transpiration, wilting occurs. This is temporary wilting. The rate of transpiration is reduced by decrease in light intensity. Temperature is the most important factor in regulation of transpiration. Plant factors that affect transpiration include number and distribution of stomata, number of stomata open, water status of the plant, canopy structure, etc. Cut flowers are dipped basally in dilute NaCl solution to reduce transpiration. Substances that inhibit transpiration by stomatal closure are called antitranspirants. Example: phenyl mercuric acetate, ABA, silicon oil, low viscosity resin. Maximum transpiration occurs in mesophytic plants. The rate of transpiration is highest when soil is wet and air is dry. Rate of transpiration is high in C3 plants. Extent of xylem sap due to transpiratory pull depends mainly on three physical properties of water: cohesion, mutual attraction between the water molecules. Cohesive forces of water is due to the H1 addition, attraction of water molecules to polar surfaces such as surface of tertiary elements. The wall of the xylem vessel made up of lignocellulose has a strong affinity for water molecules. Water column does not rupture during ascent in tertiary elements due to cohesion and addition. If cohesion and tension transpiration pull theory is correct, then break in water column should cause wilting of leaves. Surface tension. Water molecules are attracted to each other in the liquid phase more than to water in the gas phase. These three properties give water high tensile strength and ability to resist a pulling force, high capillarity, ability to rise in thin tubes. In plants, capillarity is aided by small diameter of tertiary elements, xylem tuckets and xylem vessels. Cotton fibers of blotting paper dipped in water absorb water to capillarity. Because of low concentrations of water vapor in the atmosphere as compared to substromal cavity, the intercellular spaces water diffuses into surrounding air. This creates a pull force. Transpiration facilitates absorption of water by roots. Force generated by transpiration can create a pressure sufficient to lift a xylem-sized column of water over 130 meters high. Prolonged waterlogging kills plants due to stoppage of root transpiration. Transpiration and photosynthesis are compromised. Transpiration and photosynthesis are compromised. Curtis said that transpiration is a necessary evil. Transpiration has more than one purpose. Transpiration creates transpirational pull or for absorption and transfer of water. Rate of water absorption can be increased through increased transpiration. Supplies water for PSH. Transport minerals from the soil to all parts of the plant. Cools leaf surface sometimes 10 to 15 degrees by evaporative cooling. Maintains the shape and structure of the plants by keeping cells turgid. Uh, Water movement in leaf. Evaporation from the leaf sets up a pressure gradient between the outside air and the air spaces of leaf. The gradient is transmitted into the photosynthetic cells and all the water filled xylem in the leaf vein. An actively photosynthesizing plant has an insatiable need for water. Photosynthesis is limited by availability of water, which can be swiftly depleted by transpiration. Humidity of rainforest is largely due to vast cycling of water from root to leaf to atmosphere and back to soil. The evolution of C4 photosynthetic systems is probably one of the strategies, maximizing the availability of C2 while minimizing water loss. C4 plants are twice efficient as C3 plants in terms of fixing carbon, making sugar. A C4 plant uses only half as much of water as a C3 plant for the same amount of CO2 fixed. Uptake and transport of mineral nutrition. Plants obtain their carbon and most of the O from CO2 in the atmosphere. Remaining nutritional requirements are obtained from the minerals and H2O in the soil. Uptake of minerals and ions. Unlike water, all minerals cannot be passively absorbed by roots. Two factors are responsible for it. Minerals are present in the soil as charged particles, ions which cannot move across cell membranes. The concentration of minerals in the soil is usually lower than concentration of minerals in the root. Therefore, most minerals must enter the root by active absorption into cytoplasm of epidermal cells. Active uptake of ions is partly responsible for the water potential gradient in roots and therefore the uptake of water by osmosis. Minerals absorption occurs independent of water absorption. Some ions also move in the epidermal cells passively. Thus, ions are absorbed from the soil by both passive and active transport. Transport protein in endodermal cells are control points where a plant adjusts and the quantity and types of solute that reach the xylem. The root endodermis, because of the layer of suberin, has ability to actively transfer ions in the one direction only. Mechanism of absorption of elements. Process of absorption can be demarcated into two main phases. In the first phase, an initial rapid uptake of ions into the free space or outer space of cells. Apoplas is passive. Passive absorption of minerals depend upon temperature. In the second phase of uptake, ions are taken slowly into the inner space, symplast of the cell. The entry or exit of ions to and fro from the symplast require energy, which is an active process. Lecithin functions as carrier in active ion absorption. Active increase in respiration during mineral absorption is called salt respiration. The passive movement of ions into apoplast occurs through the ion channel, the transmembrane protein that functions as selective force. The movement of ions usually is called flux. The inward movement into the cells is influx and the outer movement efflux. Translocation of mineral ions. After uptake of ions by passive or by active or passive means or by both, their further transport of the stem to all parts of the plant is through the transpiration stem. Unloading of mineral ions occurs at the fine vein endings through diffusion and active uptake by these cells. Mineral ions are frequently remobilized, particularly from molars and seedling part. Before the leaf, fall, and deciduous plant minerals are removed to the other parts. Elements most readily mobilized are N, P, and K. S and Ca are in structural composition of cell wall, therefore it is not remobilized. Much of nitrogen is carried in organic form as amino acids and related to compounds, but some of the N travels as inorganic ions. Similarly, small amounts of P and S are carried as organic phosphates. Phloem transport flow from source to sink. Food, primarily sucrose, is transported by the vascular tissue phloem from a source to a sink. Usually, the source is understood to be that part of the plant which synthesizes the food, that is the leaf, and sink, the part that needs or store the food. But the source and sink may be reversed depending on the season or the plant's needs. Sugar stored in the roots may be mobilized to become a source of the food in the early spring when the buds of the trees act as sink. They need energy for growth and development of photosynthetic apparatus. Since the source-sink relationship is variable, the direction of movement in the phloem can be upwards or downwards, that is bidirectional. This contrasts with that of the xylem, where the movement is always unidirectional. Hence, unlike one way of water flow in the transpiration, food in phloem sacs can be transported in any required direction so long as there is a source of sugar and sink able to use, store or remove the sugar. Phloem sap is mainly water and sucrose, but other sugars, hormones and amino acids are also translocated to phloem. Mass flow hypothesis or pressure flow hypothesis of Munch. The accepted mechanisms used for the translocation of sugar from source to sink is called mass flow hypothesis or pressure flow hypothesis. Sugar is first loaded into companion cells and then into living phloem sieve to cells by active transport. That loading at the source produces the hypotonic conditions in the phloem. This sets up a water potential gradient and water in the adjacent xylem moves into phloem by osmosis. As hydrostatic pressure in the phloem sieve tubes increases, the pressure and flow begins and sap moves through the phloem. At the sink, incoming sugars, sucrose, are actively transported out of the phloem, unloading. As sugar are removed, the osmotic pressure decreases in the phloem, high water potential, and water moves out of the phloem, returning to xylem. Girdling experiment is used to identify the tissue through which the food is transported. Girdling or ringing experiment or ring barking was first performed by Harding. It is in this on the trunk of a tree, a ring of bark, up to depth of phloem layer can be removed. In the absence of downward movement of food, the portion of bark above the rings on the stem becomes swollen after a few weeks. This experiment shows that phloem is responsible for transportation of food. If xylem is removed or blocked, then the wilting takes place.